All right, let's jump right into this explainer and completely demystify the science of toxicology. We're going to take these detailed, highly technical notes and transform them into a really fascinating journey, exploring exactly how your body interacts with chemicals. Now, it's a world full of hard, fast rules, but as we'll see today, it's also absolutely packed with incredible, life-altering exceptions. So, we're going to start with a crucial point straight from the source material. When we hear the phrase side effect, we almost instantly assume it's a bad thing, right? But let's rethink that for a second. A side effect is not automatically a toxic effect. It literally just means the chemical did something other than its primary intended purpose. It's an unintended consequence, sure, but definitely not necessarily a toxic one. In fact, sometimes an unintended side effect actually becomes the totally desirable therapeutic goal. Take sildenafil, for example, widely known as Viagra. It was originally studied as an antihypertensive to lower blood pressure, but it had a very specific, unintended side effect that completely changed its medical use. Or look at diphenhydramine, which is the active ingredient in Benadryl. Now, it's an antihistamine for allergies, but it causes central nervous system depression. Today, that side effect is the exact reason it's sold as an over-the-counter sleep aid. Before we get into the really wild exceptions of how our bodies react, let's just establish the baseline rules of the terminology here. We need to distinguish between what nature makes versus what humans make. If a toxic substance is produced by a living organism, like say a snake or a poisonous plant, it's a toxin. But if it's produced by human activity, like an industrial byproduct or some synthetic chemical, we call it a toxicant. It's a small distinction, but a really important one for the science. Okay. Here is our roadmap for today. 1. Welcome to toxicology. 2. When the immune system attacks. 3. Rare and unpredictable reactions. 4. Time, place, and damage. 5. The math of chemical interactions. And 6. How we build tolerance.
Let's kick things off with Section 1. Welcome to Toxicology, the baseline rules.
The foundational concept of toxicology is understanding the adverse effects of chemical or physical agents on living organisms. But here's the catch. It's not as simple as dose equals damage for everyone across the board. So why is it that you and a friend can take the exact same dose of a chemical but experience totally different effects? Well, it comes down to individual susceptibility. Our bodies just don't all absorb, metabolize, or repair damage in the exact same way. Your age, your genetics, whether you're pregnant, your biological sex, and any existing comorbidities literally rewrite how a chemical impacts you. Moving right along to Section 2, when the immune system attacks, chemical allergies. When it comes to your body's immune system, the golden rule you have to remember is this. The first exposure teaches the immune system, while the second exposure triggers the attack. The crucial point here is that most small chemicals are actually way too tiny for our immune system to even recognize. They just fly completely under the radar. But enter the hapten. A hapten is a tiny chemical that binds to your body's native proteins, and once it attaches, it forms a brand new hapten protein complex, turning it into an antigen. Suddenly, your immune system freaks out over this new combination. This is exactly what happens with a penicillin allergy. The penicillin binds to your proteins, primes your immune memory during that very first exposure, and then triggers a massive allergic response upon later exposures, even at much, much lower doses. Now, not all allergic reactions happen at the exact same speed. Let's look at the scene of the crime. Type I is your classic immediate allergic reaction driven by IgE antibodies. That hits in minutes to hours and causes things like bronchoconstriction. But then look down at type 4. This one is delayed, it's mediated by T cells, and it might not even show up for 24 to 72 hours, often presenting as a really severe, blistering contact dermatitis rash. Next up is Section 3, rare and unpredictable reactions. Idiosyncrasy. So now we're moving away from predictable allergies and classic dose-response relationships to something that's much rarer, chemical idiosyncrasy. This is based on an individual's extreme, totally abnormal sensitivity to a chemical. Unlike classic toxicity, these idiosyncratic reactions, they don't follow the rules. Most people need a high dose to show any toxicity, but these specific patients react at incredibly low doses. The source notes point to 4 distinct mechanistic failures that cause this. First, the body forms a reactive intermediate. Second, it utterly fails to detoxify that intermediate. Third, it triggers an abnormal immune response through HLA and T cells. And finally, it leads to severe cell death or tissue injury.
To put that into perspective, the incidence rate of these idiosyncratic reactions is incredibly rare. We're talking often just 1 in 10,000 patients or even less. But when they do happen, they are severe. A prime example from the notes is the drug carbamazepine. If that's given to a patient with a very specific genetic variant called HLA-B1502, it can trigger Stevens-Johnson syndrome, which is an intensely severe rash affecting the skin and mucous membranes. It's basically a perfect storm of genetics meeting chemistry.
All right, Section 4, time, place, and damage, mapping toxicity. So how do the timing of the exposure and the exact location of the chemical contact dictate the severity of the damage? Well, a single acute exposure doesn't always mean immediate damage. On one hand, we have immediate toxicity. Hydrogen cyanide, for instance, rapidly inhibits cellular function, specifically cytochrome C oxidase, which results in death within minutes. But on the other hand, we have delayed toxicity. The toxic bill for asbestos comes due months or even years later because the damage develops slowly, accumulating in the lungs to produce chronic inflammation, fibrosis, and eventually cancer. We also have to consider if the damage is permanent. Whether a toxic injury is reversible depends heavily on the target organ's own ability to regenerate itself. Organs like the liver and the GI tract have a really high regenerative capacity. This means the injury can often improve or completely disappear once the toxicant is removed. But irreversible toxicity hits organs like the central nervous system and the heart, which have incredibly low regenerative capacities. Once the damage is done there, it persists for good. Furthermore, we have to separate damage happening right at the site of contact from chemicals that travel through your circulation. Local toxicity happens exactly where the chemical touches your body. Highly water-soluble ammonia gas, for example, gets trapped in your mucus, causing intense local irritation right in the upper respiratory tract. Systemic toxicity, however, requires absorption. Carbon monoxide enters the lungs, crosses into the blood, and displaces oxygen and hemoglobin. That causes systemic, body-wide toxicity far away from the original point of contact. Let's dive into Section 5, the math of chemical interactions, when 2 + 2 = 20. Seriously, what happens when 2 or more chemicals mix inside you and just completely rewrite the expected toxic effects? Let's actually look at the math. You have basic addition where the combined effect just equals the sum, right? Like 2 + 3 = 5, which happens with 2 organophosphates. But look at synergism. 2 + 2 = 20. The combined effect is radically greater, like the massive risk jump when a tobacco smoker is also exposed to asbestos. Then there's potentiation. 0 + 2 = 10. One chemical has zero toxicity on its own, but makes another far more toxic. This is exactly how harmless grapefruit juice potentiates the effects of statins, like simvastatin. And finally, antagonism, where 4 + 0 = 1, because one chemical physically reduces or blocks the effect of another. This is the life-saving science behind using naloxone for morphine overdose. Finally, we arrive at Section 6, how we build tolerance. The body adapts. When you're repeatedly exposed to a chemical, your body fights back and adapts. It can build tolerance, meaning it responds less and less to that exact same chemical over time. You eventually need more of a drug to get the same effect. And this happens for one of 2 main reasons. First is dispositional tolerance. This means your body literally gets better at destroying or excreting the chemical before it even reaches the target site, often through changes in your metabolism. The second is cellular or tissue tolerance. The chemical reaches the target just fine, but your cells simply stop listening to it, usually through receptor desensitization or downregulation. Ethanol, or just everyday alcohol, is the perfect anchor to illustrate this. It shows how repeated exposure forces multiple bodily systems to adapt to reduce intoxication. On the dispositional side, chronic drinking upregulates an enzyme called CYP2E1, meaning your body metabolizes and burns through the alcohol much faster. On the cellular side, your neurons literally respond less because GABA signaling is reduced. And, fascinatingly, this leads to cross-tolerance. Because your body is tolerating the alcohol depressant, it actually reduces your sensitivity to entirely different central nervous system depressants, like barbiturates. So after seeing how our unique biology, our genetics, the timing of exposure, and even something as simple as your morning grapefruit juice can completely bend all the rules of toxicology, it leaves us with this final provocative thought. If the human body can adapt, overreact, or completely rewrite the mathematical equations of toxicity, is any dose ever truly predictable? It just goes to show how wonderfully complex and highly individual human biology really is.